Yo, listen up, I got a story about a teacher, Mr. Wright. Yeah, he's a biology creature. In the classroom, he's spitting knowledge like a preacher. With his lab coat on, he's the lyrical feature. He's got test tubes and beakers mixing up that potion, teaching us about cells and the process of mitosis.
Hi Year 11s, this is our last PowerPoint for the infectious disease unit of Stage 1 biology, and we're going to be looking at the acquired immune response, or the acquired immune system. So this is essentially our third line of defense. This is the specific, also sometimes called the acquired immune response, and it's activated when a pathogen has survived through our first and second line of defense, first being the barriers to prevent it from entering us, and the second being our non-specific immune system that we talked about in the last video. The specific immune system has three functions: to identify foreign materials and cells, respond to those foreign materials and cells, and to produce memory cells that recognize previous invaders to prevent them from infecting us again. It consists of highly specialized cells that identify and neutralize a specific pathogen. The cells of the specific immune response recognize molecules called antigens, antibody generators, antigens, on the surface of the pathogens. So, first off, I suppose, how do you recognize something that is a pathogen? So this is the whole idea of self and non-self. And the specific immune system, or the acquired immune system, can tell the difference between the cells of the host, self, and the invading microorganism or pathogen, non-self. And it does this by recognizing molecules called antigens on the surface of cells. Antigens are a class of materials that include proteins, polysaccharides, or glycolipids. They're present on the cells of the host and the microorganism, and they have a unique chemical structure. Foreign antigens, ones that aren't generally found in our body, will initiate the immune response. Two types of specific immunity, humoral immunity and cell-mediated immunity. Both of these are going to involve white blood cells called lymphocytes. Two different types of lymphocytes we're going to talk about are T cells and B cells. T cells are involved in cell-mediated immunity, while B cells are involved in humoral immunity. When they're inactive, lymphocytes can be found inside the lymphatic system. So now let's move on and look at each of these type of immunities. Now these, I'm not going to lie, these are probably the hardest parts of the unit. So let's start with cell-mediated immunity. In cell-mediated immunity, phagocytes engulf foreign cells, and the antigen of a pathogen will be displayed on the surface of a phagocyte once the pathogen has been digested. The phagocytes have a set of membrane proteins called the major histocompatibility complex, MHC, that selectively bind to the fragments of the digested pathogen and display those fragments. The phagocyte is now known as an antigen-presenting cell, or an APC, and the lymphocytes called helper T cells will bind to those antigens expressed on the APC. The phagocytes will then release a chemical called interleukin-1. This chemical will act as a chemical messenger and bind to the helper T cells. The helper T cells will then be stimulated to release a chemical called interleukin-2, which stimulates the growth and development of cytotoxic T cells, which are the killer T cells that we talked about last video. Killer T cells will then detect the antigen on the surface of the infected cells, and they'll release a perforin that creates holes in the membrane of the infected cell. Water and ions will then move in through the infected cell into those holes that were just made by perforin and cause the cell to burst. We then have memory T cells being synthesized that recognize the antigen of the specific infection to prevent it from being coming back again. Humoral immunity, on the other hand, involves T cells and B cells. B cells, lymphocytes, secrete proteins called antibodies. Antibodies are a special Y-shaped glycoprotein that binds to antigens on the surface of pathogens and foreign cells. Like I said before, antigen just means antibody generator. There's a series of highly coordinated steps, which you guessed it, we're going to look at now. So, 9 steps we need to know. The first one is the phagocyte engulfs a pathogen and expresses the pathogen's antigen. Helper T cells interact with those antigens expressed on the surface of the phagocytes, and the phagocytes release a chemical called interleukin-1. Helper T cells will then respond to interleukin-1 by secreting interleukin-2. Interleukin-2 will activate lymphocytes called B cells, which differentiate into two different types of cells, plasma cells and memory B cells. Plasma cells will release 2,000 antibody molecules per second, and those antibodies will bind to the antigens on the surface of pathogens, and the antibodies will inactivate pathogens through neutralization and agglutination, which we're going to talk about in a second. Those memory cells we will keep in case the infection ever comes back again. Talking about neutralization and agglutination, neutralization is when antibodies bind to the antigens on the surface of pathogens, and the binding of antibodies prevents pathogens from attaching to host cells. Phagocytes recognize the antibodies and they destroy the pathogen. In agglutination, antibodies will bind to the antigens on pathogens and clump the invaders together. The action limits the mobility of the pathogens, and the phagocytes can recognize the antibodies and destroy the pathogens through phagocytosis. So, immune response to antigens. This is just a looking at everything. So, primary response. This occurs when a pathogen first invades the body. It's the first time our body is exposed to that pathogen. It takes a few days to produce enough antibodies to kill that pathogen, which results in a delay before the host could ever overcome the disease, and hence you become sick. The secondary response involves memory cells, which are your T and B cells. It's quicker and more effective than the primary response, and if a pathogen returns to the body once you've already been infected, the secondary response is triggered. Antibodies are produced at an incredibly fast rate, and they're secreted more rapidly, and hence the secondary response can be overcome through mutations. So the secondary response can make sure you get better before you ever get sick. This response can be overcome by pathogens through mutations in the antigen, changing the shape, and then hence the antigen is no longer recognized, and the memory B cells, memory T cells, no longer work.